Hi all, I'd like to first thank the ISMB and ECCB conference for accepting our poster to present. My name is Srijan and I'll be presenting our study on fundamental limitations of foundation models in single-cell transcriptomics. Foundation models are large-scale, pre-trained models designed to capture general representations from diverse datasets. The idea is that once pre-trained, these models can be fine-tuned on smaller or different datasets to perform specific downstream tasks with minimal additional data. Thus, they reduce our need for task-specific models. These models have shown breakthrough performance in natural language processing, medical imaging, and most recently have begun being applied to single-cell transcriptomics. These models are highly promising, particularly in a field like single-cell transcriptomics, for a couple of reasons. First, they perform well on downstream tasks that have minimally available data. This is because the general field understanding derived from pre-training provides the model a starting point, so to speak, to build off of during adaptation and fine-tuning. This fine-tuned model is subsequently more generalizable than a task-specific trained model. Research on transcriptomic foundation models, or TFMs, such as SCGPT, SCMAMBA2, GeneFormer, and a myriad of others, have shown promising results in tasks such as cell type classification, gene interaction mapping, and dosage sensitivity prediction. These models leverage millions of single-cell transcriptome profiles across a range of tissues and pathologies that were given to them during training. But despite the excitement, there are a few assumptions that are concerning. First, it's assumed that the information being fed into the model is of a sufficiently high caliber that the model may learn. Secondly, it's assumed that the corpus of pre-training and fine-tuning data used is sufficiently diverse to confidently provide a sense of learning with these models, that they are truly learning the cell-gene interactions. However, real biological data is noisy, sparse, and often imbalanced. We suspect that existing models may be learning technical patterns rather than true biological context, especially when the input is distorted or unbalanced. So our core question is, do current single-cell foundation models truly learn biologically meaningful representations, or are they simply learning large-scale data patterns? To investigate this, we benchmark three prominent foundation models, GeneFormer, SCGPT, and SCMAMBA2, against a statistical baseline, Srijan v5. And we use cell type classification as our benchmark for all of these tasks. SCGPT and SCMAMBA2 both use a bin-based tokenization method, while GeneFormer uses a rank-based method. SCMAMBA2 has a state-space model backbone, while both other models have transformer backbones. We use the H. pancreas and the myeloid dataset, and their statistics are listed. A key design difference between all three models lies in the tokenization strategy. SCGPT and SCMAMBA2 use a bin-based tokenization, converting continuous expression into ranked bins. The final token E sub k is obtained by summing the gene ID token with the bin value, which is a value from an integer value from 0 to 50, and a condition token. This provides the model with relative expression information within each cell, but sacrifices gene-level resolution. Additionally, biological context is added through condition embeddings. GeneFormer, by contrast, uses a rank-based tokenization method, which selects the top-ranked genes following normalization. The process is as follows. First, each gene's count is normalized by the cell total count value and the corpus median. Then, genes are ranked by their adjusted expression values, with the top K genes being used as the token sequence input. This method better preserves relative expression signals across genes and allows the model to focus on biologically discriminative features. We hypothesized that these tokenization methods could significantly impact the performance of these models. First, we test all models on a clean, unperturbed datasets. Srijan v5, our statistical baseline, outperforms all foundation models, including GeneFormer. Among the foundation models, GeneFormer outperformed SCGPT and SCMAMBA2 by an average of roughly 5%, despite having 40% fewer parameters. However, Srijan still beat GeneFormer by roughly 9% in accuracy on average. This was our first key result. Even without noise, the foundation models underperform. To stress test the robustness of both tokenization methods and all three models, we added Gaussian noise to the gene expression matrix before tokenization in order to amplify any effects of noise that arise post-tokenization. Doing this amplifies any distortions introduced by the tokenization process itself. On the myeloid dataset, SCGPT dropped roughly 35% in accuracy at just 20% noise. GeneFormer, using rank-based encoding, dropped only roughly 25%. On the H-pancreas dataset, the pattern was even more stark. SCGPT dropped nearly 59% at 30% noise, while GeneFormer only dropped 25%. This showed how bin-based encoding was highly sensitive to noise, while rank-based methods were more robust. Next, we explored how class imbalance affected predictions. In myeloid, rare cell types were underrepresented, while CD14 monocytes were dominant. SCGPT overpredicted CD14 cells by roughly 16%, heavily skewing class distribution. SCMAMBA2 showed 100% overprediction. GeneFormer was more stable, with roughly 6% overprediction, and achieved 26% higher macro F1 than SCGPT. This suggested that foundation models were highly sensitive to sampling bias, particularly when using binning. There are a few lessons to be learned of the limitations of these foundation models we assessed. First is that current tokenization methods lack sufficient information of biological state to allow for the understanding of the context and the cell as a whole. This can be shown by the noise experiment conducted, in which GeneFormer's rank value encoding preserves biological context via global gene normalization and performs far better than the bin-based methods that both SCGPT and SCMAMBA2 use. As a result, incorporating biological context, such as cell ontology, may improve model performance. This has already been conducted with SCCello. Thank you for listening. I'd be happy to answer any questions or regarding the experimental setup. Simply contact me at srijan.atti@gmail.com or drsubramaniam@shankar@ucsd.edu.